Scientists may have just found a way to slow down reproductive aging. Here's what that means and why it could be good news for anyone who wants to have children later in life. It's me, and this is MonsXP. The ability to reproduce is a defining characteristic of living things, but being able to do so successfully requires a lot of different parts to work together in just the right ways. And as we age, a lot of those parts wear out or get used up, resulting in reproductive aging, which can start at different times for different organisms. In humans, reproductive aging is associated with a decline in the number and quality of eggs a person has, and also with changes in the uterine lining that make it less hospitable for an embryo to implant. All of these changes are driven by our genes, but exactly how those genes control reproductive aging has been a mystery. And this is important, because if we understand the mechanisms of reproductive aging, we might be able to intervene and help people maintain their fertility for longer. So scientists have been searching for the key genes and the clues to slowing down reproductive aging. And in 2023, a group of researchers uncovered one piece of that puzzle. They identified a single molecule that seems to play a critical role in reproductive aging in mice. And, importantly, they found a way to slow down that molecule and thereby slow down reproductive aging. To find this aging-related molecule, the team took a closer look at the ovaries of young and old mice. They noticed that in old mice, the cells in the ovaries had more of certain kinds of molecules called AGEs, or advanced glycation end products. AGEs form when proteins bind to sugars, and they can build up inside and outside of cells. And while AGEs are found in a wide variety of tissues and generally associated with aging, nobody had ever looked at them in the ovaries before, and nobody had ever demonstrated that they actually contribute to reproductive aging. But something about the ovaries of older mice just didn't seem right to the team. The ovaries were full of AGEs, but they weren't evenly distributed. Instead, they seemed to be concentrated around the blood vessels. And upon closer investigation, the team realized that the AGEs were actually stuck on the surface of a type of cell that lines the blood vessels. These cells, called endothelial cells, normally help eggs move from the ovary to the uterus. But when the team looked at the ovarian endothelial cells from older mice, they noticed that they were coated with a thick layer of AGEs. And the thicker the coating, the older the mouse. Since nobody had ever linked AGEs to reproductive aging, the team needed to figure out whether the high levels of AGEs they observed in old mice were actually causing reproductive aging or were just a side effect of it. To do that, they injected some of the aged ovarian endothelial cells into young mice. And sure enough, the young mice that received the old endothelial cells began to age faster. Their ovarian endothelial cells accumulated more AGEs, and their fertility declined more quickly than the young mice that received endothelial cells from other young mice. This showed that AGEs on the endothelial cells were actively contributing to reproductive aging. But why? The team suspected that the AGEs were interfering with the function of the endothelial cells, but they weren't sure how. After all, the endothelial cells of old mice still seem to be doing the critical tasks of transporting nutrients and helping eggs develop. So the team dug deeper and discovered that the AGEs were preventing the endothelial cells from contracting properly. Normally, endothelial cells line blood vessels throughout the body, including the ovaries, and they help to keep things moving smoothly. But when the endothelial cells of the reproductive system encounter AGEs, they can't contract, which prevents the smooth transport of eggs and other essential molecules. When the researchers inhibited age formation in old mice, the mice's reproductive systems worked better. Their eggs developed normally, and they were able to get pregnant at higher rates. This suggests that slowing down age accumulation could potentially slow down reproductive aging. And while this research was done in mice, reproductive aging is a universal process, and these findings could apply to other species as well. By shedding light on the role of AGEs in reproductive aging, the study uncovered a potential new avenue for interventions that could help preserve fertility for longer. Maybe one day soon we'll be able to slow down reproductive aging too.